If Earth is about 4.5 billion years old,
Yeah, let's give it the universe. I'll give you 14 billion.
old is 1 billion years not enough time for randomness?
No, not at all. And
Your positions, are you giving all of this evidence to say that the evolution theory is not true, cannot be true?
James Tour recently spoke at Harvard University, and these Harvard students press him on the origins of life and how the world began. So let's get right into it. Shout out to James Tour. You can check out this entire video on his channel. Let's get into it.
So particular that you can only do it in the lab, but not on Earth.
no, there's enough energy out there to do this. So the problem is you want to hook them together in particular ways. You have to hook them together in particular ways. And they end up hooking at the wrong sites. That's what happens. And so in the lab, what you do is you take human-designed groups and you block all the other positions, and then you hook just the two remaining positions, and then you deblock them. So it's a very sophisticated human-derived thing with all these human-derived methodologies. And so it's like each one of these steps is like building a car. I mean, it's a lot of work that has gone into developing these methods. So you can do it in a lab using modern techniques, but they're not prebiotically relevant, meaning that it could have never happened in nature that way because all of these are human-designed methods. Yeah, but there's plenty of energy to do this. Yeah.
Hello. Thank you for your presentation. You mentioned the concept of prebiotically relevant a few times. I was just wondering what you meant by that. And I was curious as to your positions. Are you giving all of this evidence to say that the evolution theory is not true, cannot be true? I wasn't able to grasp your position on this fully.
So prebiotically relevant means that you have to use conditions that would have been available on an early Earth. So the early Earth didn't have laboratories. The early Earth didn't have many human-made reagents that we buy from chemical manufacturers. So you have to do things mimicking those conditions. That means it's prebiotically relevant, right? So what we do in our laboratories today is not prebiotically relevant because we use techniques and we use tools that were not available on an early Earth. Okay? That's what prebiotic relevant means. Now, I didn't touch at all on evolution. So there's two different areas. There's abiogenesis. There's the study about how life came about. And then there's another area of evolution. Once you have life, how do you go from simple forms of life to more complex forms of life? That's the study of evolution. I never touched on that. I only dealt with how do you make the first life to begin with? Because, and so Charles Darwin never dealt with abiogenesis. He even conceded that that's an unknown. That was an unknown for him. Evolution is something else, and that I didn't deal with. Now, evolution has its own problems. There's a lot of problems with evolution. But that's a whole different study. But I didn't even touch on that.
Thank you, Dr.
Yeah, I think that's very interesting because a lot of atheists or people who don't believe in God are like, you see, evolution debunks God. Evolution debunks the creation story. Well, does it? Because he's said right there, it doesn't even tackle the origins of life, how things came to in the first place.
For your beautiful prayer. I wanted to ask you also on this topic of abiogenesis and the chemistry about if it's possible to derive some of the more complex structures out of a simpler subset of original structures, thinking about whether, for example, in the area of amino acids and proteins, if for some of the proteins that have multiple functional places for attachment, whether it's possible for the subset of the 8 amino acids that don't have such points of attachment, if they could potentially evolve out the other amino acids, and in particular, whether it would be possible to, in more controlled environments, like, for example, the deep sea vents hypothesis is one that gets kicked around a lot in the abiogenesis circles, or there's also the sort of deep sea pressure pools hypothesis, which is somewhat distinct from the deep sea vents, because that's also low temperature, which facilitates far long, and high pressure, which facilitates far longer half-lives for the molecules at play on the bottom of the ocean.
Okay, so first of all, deep sea vents, which are hot, the lifetime of molecules is much shorter, okay? Not longer, much shorter. And the deep sea vents are at temperatures where you get racemization, meaning that if you happen to have something where you had molecules with pure stereogenicity, meaning that they were enantiopure, they had one-handedness, those are now going to racemize at those higher temperatures. Racemization is always faster than coupling reactions. It's another problem with the deep sea vent hypothesis. If you deal with the 10 amino acids that don't have active side chains, and you say you could make polypeptides from those, you could. But without those reactive side chains, you don't have the ability to do the folding that you need to fold. Could those somehow then form a template that give you the other ones that might help to make the other ones? You could do that. It'd be hard to come up with a scenario, to map out a scenario, but if you're just thinking wildly, yes, the answer is yes. Could you make enzymes? But we don't know of enzymes that don't have any of the ones with the active side chains. Those are generally the ones that give us the strong interactions for other groups to hold them in place. The weaker interactions do come from those other groups. You get these hydrophobic interactions. So that's an interesting hypothesis. And so what I always do is I give you that. I'll just give it to you, even though it'd be hard to think about that, but I'll give you all the polypeptides you want. Let's just say they can all be made. Now what? Remember, I've given you the entire deconstructed cell with all the correct all the correct polymerization. So what you're going to make is random, and what I'm giving you is with a code. Now what? You've still got to, and I'll give you all the sugars, even with all the code. Even if you had all of those, we don't even know what to do. We have no idea how to package these things together. We got to deal with an interactome that has 10 to the 79 billion combinations. You know, wow. So many hurdles. So what I do is it's sort of like what my father did. My father used to give Christmas presents to everybody, and at Christmas time he would give $1 million gifts. He would buy raffle tickets and give them to people. He was very generous with this. I'll give it to you. This is what I do. I'm very generous with giving whatever part people want. I give it to them.
Yeah. In that sense, you have out of 100 million or something that this possibly could happen, but very, 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 very unlikely.
Because then they see, there's this forest of other stuff that I've got to deal with that makes this impossible. Okay.
Thank you for your talk. I was thinking that when we think about DNA, RNA, and protein, the one thing that comes to my mind is structure. Do you think structure was evolutionarily conserved when we talk about evolution or any kind of model?
Yes. Structure is always conserved in evolution, always conserved. If it were not, there would be no evolution. Structure has to be conserved, and you see that all the time. I mean, you can watch evolution in a laboratory. This microevolution, as it's called. I mean, we do a lot of bacterial work in my own lab. And you can see changes in bacteria that occur, but structure is always conserved. If it were not well conserved, there would be no evolution. In fact, one might even be able to put as one of the definitions of evolution as a structural conservation with small perturbation.
So is this person back here sleeping? Seriously? While he's having this conversation? We just have a girl sleeping in the back. Wow.
Always have such compelling stories and data. So I hear you when you say that like scientists are like always almost discovering the answer to this, but we're really finding that things are only getting more and more complicated the more knowledge and information you find out. But then you also had that point that's like science has exponentially advanced to the degree that we're able to find answers to things that we never would have imagined in the 1700s. So I guess my question is, let's say science does discover the origin of life, like where would that leave you and your initial proposition for this talk?
I would be so happy. I would be so happy because, prior to the mid-1950s, people did not really understand a molecular basis for the fact that if you have two parents that are tall, their child is tall. They didn't have a molecular basis for that. And then the structure of DNA was figured out just in like 1952, 1953, right around in there. So certainly within the lifetime of many people that are alive today, certainly in the lifetime, earlier in their life, it wasn't known. So that doesn't make God less in my eyes because we understand how he preserves this information. That actually makes him more marvelous in my eyes. It's like, oh, that's how you did it. I mean, this is amazing. And there's this whole molecular basis for this. And that's why I'm a chemist. I mean, God's a chemist. I mean, it's all stored in the molecules like this. So I think the more we learn about creation, the more amazing God becomes. Because it's not just a bunch of protoplasm that does this.
Yeah.
It's such a sophisticated system that does this. So what we're learning about cellular structure, for example, is that there's layers and layers and layers of information that have to take place here. And it's like this massive computer program of layers of information and this whole area of systems chemistry that needs to take place. It's really, really magnificent. I say as a scientist, I chase the universe trying to explain it. And God's word defines the universe. He says something, and these amazing things happen. And so it actually makes me able to appreciate far more, I think, creation than the man or the woman on the street can appreciate it. Because I can look at a leaf and in my mind's eye, this is what I do. I'm projecting in my mind's eye. I look at that leaf and I think there's a magnesium atom sitting in the middle of a porphyrin. So these nitrogen atoms surrounding this magnesium. And there's photons of light hitting that, and electron injection now occurring, down 100 angstroms occurring to start the photosynthesis process to convert CO2 to carbon to build that tree.
That's so amazing. The more you know, the more you become more and more in awe of God, I would say. And just because we find out how the origin of life began, or like the process, I guess, like we can understand maybe, how in the world would that say God is debunked then?
Who else can do that? I mean, there's not many of us that can do that. Now, if you've taken organic chemistry, you can do that. So it gives you a much greater appreciation for the wonderfulness of God's creation. It doesn't make God less in my eyes. It makes him even greater.
Dr. Tor, you mentioned that time is not your friend, and that there's not enough time for these structures to form during randomness. Just so I understand the time scale, if Earth is about 4.5 billion years old.
Yeah, let's give it the universe. I'll give you 14 billion.
Okay. And life around 3.5 billion years old, is 1 billion years not enough time for randomness?
No, not at all. And in fact, it's not 1 billion because we know that nothing could have happened before the cooling from the Great Heavy Bombardment. So after the Earth cooled, then these things could start happening. The Earth was just blazing hot. And this is why we have all these elements on the surface of the Earth. It's amazing. We have the periodic table right here. All of these is just showered here. And so we have so much. And so it was after that cooled, and it turns out that the time between the Earth cooling and the signs of life is very, very short. It's very short. And there's no way that the time, so the time is actually not 14 billion years, it's not 3.5 billion years, it's not even 1 billion years. It's something on the order of hundreds of millions, which is way, way too short for having life. And, but also—
Why is she smiling? Is she like, it makes sense? Or like, I don't believe what you say?
just molecular stability. It's not just too short for life to have formed. It's just molecules don't stay around. Like I gave you this example. If you happen to have a sugar on which there is a base, and you happen to have 4 different bases on the different sugars, and they happen to have the right stereochemistry, all this is mountains of— that's hard to do in a modern laboratory. And then, if those somehow hooked together to form a code that could code for something, the building of an enzyme that could end up building some other structure. That RNA only lasts 4 hours or less, because it's probably longer. So it's not even now hundreds of millions of years. You only got 4 hours. And now it's got to find all the amino acids in that 4 hours. So it's like, there's only moments. There's only moments. And so if you could meet an organic chemist someday, meet them. There's none in this institution, apparently. But if you could meet one of them, you would say, well, what happens if you just don't work up your reactions? You just go home for the night and go to sleep and come back. They go, oh, no, I won't do that. I tried that once, and my reaction was just toast by the next day. These things don't last. They fall apart. And that's what nature is beset with. And they hydrolyze. And we have this atmosphere, this earth that's filled up with water. And water hydrolyzes a lot of these structures quite rapidly.
That's really interesting to think about, because he understands on a molecular level, as being an organic chemist, how complicated it would take for these molecules, these sugars, these enzymes to code or to form or to come together before it just falls apart. That's wild.
Hi, doctor. Thanks for your time. When I'm talking with atheists, usually the conversation they start with, like, what's your best evidence for the existence of God? And usually one of the places I start is life itself, consciousness, the fact that you wake up, there's trees and people, etc. And so therefore it begs the question, who created all of this? And so the conversation usually goes towards like, well, if there is a God, there is a creator who created that God. And you kind of get into this infinite regress problem. So I'm just curious if I'm sure you've addressed this before. I'm just kind of curious to hear what your response typically is.
Yeah, actually, I've never been asked that before. But I've heard people talk about this argument, but people don't normally ask me philosophical questions like that. So God is an uncreated being. He's always been. And that would be the typical answer, which is the biblical answer to this. And these are hard questions. There's something as fundamental as where did life come from? Just even life, let alone consciousness. Although there is an area now within biology that is talking about the conscious-like behavior of a cell. Because it responds. You poke a cell and it learns, don't go there, I get poked. It learns how to deal with things, and bacteria are always changing based on what they're confronted with. And so it's almost as if they have some sort of cognition going on. It's different than the consciousness of a human being. But these are more than just simple little sensors that, oh, that is a molecule. There's something more sophisticated, so that interaction causes this huge cascade to occur within a cell. So a cell now develops so that an efflux pump to protect a bacterium so that it forces a drug back out. I mean, all these changes have now occurred based on an influence that came into it, which is approaching what consciousness does. We get confronted with something, and now we come with some response that we never would have come with had that influence not come upon us. So that we're seeing, even in the simplest of life forms, you see these sorts of things, and you go, wow, this thing is amazing.
That's cool.
How does all this come about? There is a reason why we build robots out of silicon and plastic and rubber and wires. Why don't we just build robots out of molecules? Like we're built. I mean, we have all these ubiquitous examples that we can do this. Why don't we just build out of molecules? It apparently works. Because we have no clue how to take molecules from a flask and build one of us. No clue. So we rather build it out of silicon and all these other materials and just kind of make it look like one of us. There's a reason for this. Molecules are like the hardest thing that you could think of to build sophisticated structure out of. It's so hard to do this with molecules. I'm not sure I answered your question, but okay.
Wow.
Hi, Dr. Tor. Thank you for your time. I consider myself a Christian, but one of the issues I find is like when I read the Bible, I want it to be very consistent with what we see in the world, like based on science. For me, like for example, when I hear in the Bible like the earth is 6,000 years old, or like, you know, God literally took Adam's flesh and took it away to create Eve, I find it like hard to believe. Either it's like we have to believe some sort of metaphorical meaning of the Bible, or are we defending like the actual words on the Bible that says he did that? So what's your sort of response for like believing in the Bible and in the science?
Well, I never read in the Bible that the earth was 6,000 years old. So that's an extrapolation that you got from people counting lifetimes back.
Yep. I asked him this question. It's so funny. I got to interview him on a Zoom call, but I asked him this question. I'm interested to see what he says here.
Of which we know that there are certain gaps there. But even if they weren't, let's just say you counted back. The Bible, to me, seems pretty clear somewhere between Genesis 1, chapter 1, verse 1, and Genesis chapter 1, verse 3, something happened. And there was a void time. And with that void time, we have no idea what the age of the earth is. Right? Yep. So we have no idea anymore. We have no idea from that. But what we do know is throughout the 1950s, so this is in my lifetime now, throughout the 1950s, most scientists believed that the universe had no beginning. It always was, the steady state approximation. And then now these proposals that it had a beginning came much earlier in the 1920s, but it wasn't until 1964 with the discovery of background microwave radiation that we realized that the earth the universe had a beginning. It had a beginning.
And all along, the Bible was right. In the beginning, God created the heavens and the earth, which addresses saying that there was a beginning.
And we were in an expanding universe, and it was confirmed in 1964. And so the whole mindset shifted. So science changes dramatically over periods of decades. It changes. What killed off the dinosaurs? It was believed that climate change. Well, no. The Alvarez hypothesis, 1980, says that an asteroid hit the Yucatan Peninsula, iridium-rich, filled the earth, filled the atmosphere with dirt and soot, and that killed off then the plants, killed off the herbivores, which killed off the carnivores, and that was the end of them. So science is constantly changing in its views. I mean, even more recently, what was formerly junk DNA when I started my career is now intergenic DNA and turns out to be super important for all regulation. Mm-hmm. And so now we're nowhere close to being similar to a chimpanzee. We were told we were 98% the same as a chimpanzee.
Wow.
Well, that's only for the protein coding part. When you look at the regulatory part, we're very different. So science is always changing. And our interpretations of the Bible change. And so how do we interpret these things? My interpretation of the Bible is constantly changing throughout my lifetime. I get different interpretations. So I've never seen a controversy between a fact of science and something that is clearly stated in the Bible. I've never seen it. If you've seen it, share it with me. I've never seen it. There are scientific theories But theories are changing all the time. There are interpretations which you can have on a Bible portion, but another person can have another interpretation. There are other portions of the Bible that are not nearly as subject to interpretation because they're said multiple times over and over again. Touch me and see, I have flesh and bones, as you see, I have. And he just walks us through it, puts his hands on our cheeks and says, you hear me? Those things are very clear. And so you have both aspects of those changing. But, there's some things in science that will never change. Water is H2O, 2 hydrogens and an oxygen. You can have different isotopes. Anywhere you go in the universe, that's going to be the same. But there are other things that are sometimes taught as facts that are just theories. So I haven't seen the controversies, personally.
Very cool. All right. That's absolutely amazing. Shout out to James too. If you want to watch the rest of the interviews and the presentation, go check out his channel. It's called The Origin of Life Crisis. So guys, I encourage you all to go to Jesus today. Know that he loves you, he's longing to have a relationship with you. Put him first. It will be the best decision you guys could ever make.